Hey everyone, my name is Meg Kilgore, and today we're going to be talking about idiosyncratic nutrient requirements of cats, which appear to be diet-induced evolutionary adaptations. So, first and foremost, we have to figure out where cats are lacking. So, why do cats need such high protein content? This is because there's not any regulation of aminotransferases. All of the nitrogen is being dispensed out of the system through ammonia and into the urea cycle. And then there's not any regulation on the urea cycle enzymes. And then some amino acids we're going to be looking at is taurine and arginine. And then some of the vitamins that we're going to be looking at is calciferol, niacin, and retinol. And this picture is just representing where amino acids come in and where nitrogen goes. When during an aminotransferase reaction. So as we can see, it gets put onto glutamate, and because this isn't happening, most of the reactions are getting rid of the amino group and putting it into the urea system. Okay, so first and foremost, aminotransferases. Um, as we can see, glutamate can go to alpha ketoglutarate, and alpha ketoglutarate can go to glutamate, and this is through glutamate dehydrogenase or glutamine synthetase. So, looking at this, we know cats are carnivores and that they have a higher protein requirement, which could be due to a higher requirement of nitrogen. So when cats were given high and low crude protein diets, there wasn't much change in aminotransferase activity. So the nitrogen was not being converted into dietary protein like it was in the omnivores tested. So as we can see here, the amino group from glutamate goes out to urea. So it's not being recycled and put back on the glutamine. It's out of the system, so it's not being used, which isn't good if you're trying to make more amino acids. And then this picture is just showing a more close-up picture of the glutamate dehydrogenase reaction, and just so you can see that the ammonia group is leaving and alpha ketoglutarate is made. So the urea cycle occurs in the mitochondrial matrix and the cytosol, as you can see in this picture on the left. It also occurs in basically 5 enzymatic steps, as you can see in this picture on the right. So typically omnivores, when they get a diet with a higher protein content, it typically correlates positively with the amount of nitrogen being synthesized into ammonia. And the opposite is true for a diet with lower protein content. So the omnivore will conserve the nitrogen to make dispensable amino acids. So the nitrogen, instead of being flushed out through urea, it's being reuptaken into the system to make more amino acids. However, when cats were given a low protein diet, there was not any decreased activity of the urea cycle enzymes. So pretty much all of the nitrogen is being lost to ammonia instead of being reuptaken to make more amino acids that they are not getting from their low protein diets like the omnivores do. So this brings us to our first quiz question, and that is, because of the lack of regulation of these enzymes, amino acids are more readily available to go into gluconeogenesis. So I want to know what that would mean for glucose levels during starvation, and I would like you to use 1 to 3 sentences to answer that question for me. Now that we have talked about all the enzymes and amino acid degradation, I want to talk about the specific amino acids that are necessary in a cat's diet that might not be essential amino acids overall. When studying the essential amino acids, the authors of this study would eliminate one amino acid from the diet and study the weight loss and just overall clinical signs on kittens. So when arginine was taken out of the diet, the kitten experienced drastic weight loss overnight, as opposed to the days it would take for the kitten to experience that drastic of a weight loss when one of the other amino acids was taken out of the diet. As I'm sure you recall, arginine is not typically an essential amino acid. However, in kittens, it is essential. So, synthesized arginine is thought to come from the urea cycle, but in kittens, the hepatic systolic arginase is overactive, and this enzyme catalyzes the degradation of arginine into ornithine and urea. So as you can see, I just pulled this close up from the urea cycle so you can see how arginine goes into ornithine and urea is a byproduct of that. And however, glutamate and glutamine can produce citrulline, which can be converted into arginine in the kidneys. And that is shown in this picture over here from the paper. So as you can see, P5C is a very important intermediate in this, and it goes all the way down into ornithine, which again we have in the urea cycle, and then ornithine can be converted into citrulline. Now, citrulline can be converted into arginine in the kidneys, and because the kidneys do not use a whole lot of arginine, it is shipped around the body to wherever it is needed, like the liver, for example. Okay, so now we're going to be talking about hyperammonemia. So kittens receiving the arginine-free diet started exhibiting life-threatening symptoms within an hour. So what caused this to happen? In growing kittens, there is an extremely low activity of the P5C synthase enzyme, which is right here, and the ornithine aminotransferase enzyme, which is right here. So what does this result in? So citrulline is not being made to form more arginine. So because there's no arginine coming in from the diet, the kittens are completely devoid of it. So the liver is the primary organ with all these urea cycle enzymes, and in cats, the arginosuccinate synthetase has such low activity rate that limits the formation of urea. So because we're in the liver, we're not making enough urea, we have such a buildup of ammonium that is causing this ammonia toxicity from the degradation of glutamine and glutamate. So kittens who have arginine-free diets but were supplemented with citrulline and ornithine did okay. The ones with citrulline were perfectly healthy. However, the ones that had ornithine substituted did not grow at all. And this is because ornithine does not completely substitute arginine. It only helps with the removal of ammonia. And if you want to see that more closely, you can click back to the last slide where you can see that the formation of ornithine helps remove urea. Our next amino acid that is essential for cats is taurine. It is a beta-sulfonic acid, which means that it is not occurring in polypeptides. Taurine is a free amino acid in animal tissues, and this amino acid goes to make bile salts, but when cats were given a taurine-free diet, it led to cardiomyopathy, retinal degeneration, reproductive problems in the queens or the female cats, and developmental problems in kittens. So, taurine forms bile salts in cats and dogs, but other animals can substitute this taurine for glycine. However, the hepatic CoA amino acid N-acyltransferase enzyme is what determines the amino acid that can be used to form bile salts, and it is thought to have a low affinity for glycine in cats, hence the obligate need for taurine. So taurine is synthesized from the oxidation of cysteine. The activities of these two enzymes, cysteine deoxygenase and cysteine sulfonic acid decarboxylase, have relatively low activities, and because of this, there is virtually no traffic along this pathway, hence the reason that taurine is essential. So all of this brings us to our next quiz question. So this is one pathway in which cysteine is used, but what does the other pathway containing cysteine make? I'll give you a hint. It's in chapter 22, and I want to know what the product at the end of the pathway is used for. And I want that in 2 to 4 sentences. Now that we have talked about the essential amino acids and the activities of a lot of different enzymes, we are going to go over a little bit of the vitamin differences in cats. Okay, so this is just a short overview of all the vitamin differences. So in vitamin A in cats, it's not synthesized from carotenoids, and there's lots of vitamin A in cat foods that can lead to vitamin A toxicity and malformations in kittens. For vitamin D, it is activated from the UV rays that come from the sun. So to activate the vitamin, cats tend to lay belly up in the sun because their hair is thinner on their abdomens as a way to absorb it into the skin. And cats also have a low concentration of 7-dehydrocholesterol in their skin. For niacin, cats have all the enzymes in the pathway to form niacin. However, the picolinate carboxylase activity is extremely high. So coat color in cats is reliant on melanin, just like skin color in humans. And there are 2 types of melanin, which are copolymers, meaning that they can be codominant or occurring at the same time. And these copolymers are cysteine containing pheomelanin or reddish brown, like we see in Peter, or eumelanin, blackish brown, like we see in Bellatrix. So because they are copolymers, they can be codominant, which means they can occur at the same time, like we see in Susan the Calico or Loki the Tuxedo Cat. And keeping hair color in cats is reliant on how much phenylalanine and tyrosine is in the diet. Moving on to fatty acids, cats cannot synthesize arachidonic acid from linoleic acid very efficiently. This is thought to be caused from the low desaturase content in the cat's liver. As you can see here, there's a desaturase enzyme and there's a desaturase enzyme. So there's 2 desaturase enzymes needed just to make arachidonic acid. So cats not having very many of these enzymes would cause the inefficiency of this pathway. But animal fat has a high concentration of arachidonic acid, so as long as they're eating animal fat, they're pretty set. The cats will also be okay if they're giving linoleic acid in their diets from plants, but it did have some negative effects for females than males because the females' litters had low capabilities of handling certain fatty acids, and the males did perfectly fine.